When they wheeled the aluminum gurney into our cold room on November 14th, 1995, I thought it was a joke, a prop for some late-night prank, something to laugh about over stale coffee. Then the lid came off the transport crate, and I saw how wrong I was. The carcass wasn't a bear or a deformed elk. It wasn't any animal in our reference folders. It was too upright in the shoulders, too long in the forearm. The hands, if you could call them that, had opposable thumbs and nails, not claws. The hair was coarse, dark, and lay in a pattern that suggested cortical musculature beneath. There was a face in there, but it had no human smile and no ape snout either. It sat like a question in our lab, all sterile tile and humming freezers, waiting for someone to answer it. I should say, before the story bleeds into legend, that I am a molecular biologist by trade. I ran the sequencing core at the Northwest Institute for Comparative Genomics. For 23 years, I'd learned how to make a sample tell the truth, provided the sample was honest. I knew how to strip away superstition and leave facts on the table, read depth, base calls, banding on a karyotype. Data was my lingua franca. That night, data would turn my life inside out. When the first sequencing reads scrolled across my monitor, I did what any careful scientist does. I cursed. Not because the numbers were wrong, but because they refused to fit into tidy boxes we kept for living things. The software flashed a similarity score against our human reference, 98.7%. I watched that number like a man watching a cliff appear where there had been flat land. I refreshed the alignment twice, then manually inspected several loci. The reads were clean enough. The coverage was shallow in places, the sample had been field preserved in a way that suggested haste, but not degraded in the pattern of old tissue. That number, 98.7%, is part of what makes the rest of this account plausible and also dangerous. In my analysis, based on the methods and references I used that weekend, the genome-wide similarity to Homo sapiens registered at roughly that figure. I say in my analysis because this is my record of what we saw and how I interpreted it at the time. I cannot and do not present it here as incontrovertible proof for the world at large. The ratio, however, changed everything in our room. If a stranger is reading this and thinking in headlines, Bigfoot, remember that our responsibility then, as now in this telling, was not to sensationalize, but to question, to measure, to model. The screen did the measuring. The rest of the night was people arguing with their instruments. By process, we logged the specimen into our laboratory management system at 2142. The transporter left with only a slip of paper. Anonymous donor, perishable biological material, with a note, chain of custody, pending. Patricia Walsh, our institute director, insisted on strict logs. She and I took the first set of swabs. We worked inside a negative pressure hood. The lights in the room were a thin, functional white. My hands ached from nitrile gloves and the cold. We sampled muscle from the left deltoid, 3 hair follicles, a skin punch from the upper thigh, and a small fragment of cartilage from behind the ear. The goal was redundancy: tissue for DNA, tissue for histology, hair shafts for mitochondrial analysis. I kept a sterile blade on the tray. We photographed everything without showing anything more than a silhouette in the record. We were scientists, but not fools. The first thing we did, as we always do, was a smear and a quick cytogenetic prep. For that, you take cells, arrest them in metaphase, and stain the chromosomes to count and look for gross structure. I set up the centrifuge. Patricia ran the camera for the karyotyping station. We cultured fibroblasts overnight, made the mitotic arrest with colchicine, and the next morning I pulled slides under a microscope. The metaphase spreads were messy by human standards, but legible. Not 46. The spread settled like a constellation under the lens, about 48 chromosomes. 48. I said it aloud because numbers need sound to be believed. 48 is not a trivial aberration. Humans have 46 chromosomes. Chimpanzees, gorillas, and orangutans have 48. Chromosome count alone does not define species. It's a clue. It suggests a divergent karyotypic history, a different set of chromosomal fusions or rearrangements somewhere on the evolutionary tree. Again, this was my observation, not a proclamation. I reported it to Patricia and to the internal log with the clinical detachment we've all trained into our voices, but in the back of my throat there was a question that tasted like lead. If this was real, where was it from? I rang a colleague across the hall to run a quick PCR on a mitochondrial marker while the culturing continued. Mitochondrial DNA is small, maternally inherited, and tends to survive harsh conditions longer than nuclear DNA. The sequence we amplified matched primate motifs in the database, but clustered outside the typical human haplotypes. It was closest to human sequences, but clearly distinct, a matrilineal line that had separated from the human mitochondrial pool long ago in my assessment. That told me two things at once. The specimen was related to us, and its female lineage had been on its own for a long time. Sequencing proceeded in fits and starts. We did Sanger reads on key loci and shotgun libraries for broader context. The alignment to the human reference genome returned the 98.7% similarity. Genome lied, the base pair matches were overwhelming but not complete. Where it differed in ways that suggested not noise but divergence. Those differences weren't concentrated in some mystery gene, but spread across non-coding regions and certain exons that influence cranial development and limb morphology.
Again, in my hands and with my equipment, that was what the data said. From those differences, one can make models of divergence time. This is standard molecular clock reasoning. Mutations accrue at an average rate, and by comparing genetic distances, you can estimate when two lineages last shared a common ancestor. With the loci we had, and applying the conservative rate constants we use for hominin comparisons, the model put the split at about 1.2 million years ago. That figure felt absurdly large, and, simultaneously, chillingly close. If the model was even approximately right, it meant there had been, on this continent, a lineage separate from modern humans for more than 1 million years. I put the qualifier in my lab notes. Estimate, model dependent. A model is just a story with math. It helps you imagine timelines, but it warns you too, with its confidence intervals and its asterisks. There were other, more practical concerns. We probed immune loci, the genes that determine how an organism recognizes and responds to pathogens. In humans, we look at HLA diversity because it confers population-level resilience to disease. The sample's immune gene region showed low heterozygosity, by my calculations, markedly lower than the global human average. That suggests a narrow pool of alleles and a population that had gone through genetic bottlenecks. The consequence is straightforward: susceptibility. A species with little immune diversity is ill-equipped to withstand novel pathogens, like those carried by humans moving into their habitat, or vice versa. We ran telomere assays too. Telomeres are protective caps at the ends of chromosomes that shorten each time a cell divides. They are a rough biomarker of cellular age and replicative history. The telomeres we measured were shortened beyond what we'd expect for a wild animal of its apparent size and development. Shortened telomeres can reflect stress, small breeding populations, or a lifetime smoked with infection and resource scarcity. In my clinical phrasing, the telomere profile suggested inbreeding and a population that had been pushed toward genetic exhaustion. My models, which I ran on the back of printouts while the fluorescent lights hummed, suggested a small effective population size. Effective population size is a term from population genetics. It represents the number of breeding individuals that contribute to genetic diversity. The numbers I used placed the effective size at a level that in established models forecast decline and eventual loss without intervention. When I say forecasts, I am saying models indicate, and with both those words I mean to be as careful as a man who knows how much gets lost in words. Models are not oracles. They are, however, enough to set a room on fire. If the science reads cold, the room did not. There was a moral temperature to the air that night, heat generated by fear rather than breath.
The implications multiplied. A hominin-like lineage in our backyard, a population on the verge of collapse, an immune naivety that could make them casualties in our wake. Publicity could bring scientists, conservationists, opportunists, tourists, and worse. A published paper could be a map and a sentence that read, in effect, "Here they are, come take them." The instinct to publish is so ingrained in us that we sometimes forget it is a kind of violence. Knowledge can be a tool. It can also be a spear. Patricia understood the stakes quicker than anyone in the building. She called an emergency meeting in the small conference room where the coffee machine hadn't been B-scaled in months. Present were the Institute's legal advisor, a senior epidemiologist friend I later learned had ties to the CDC, and 2 security officers who wore their boredom like armor. We laid our results on the table: the karyotype printouts, the alignment screenshots, my telomere graphs. The look around the table shifted. What had started as curiosity had snapped into urgency. "These are preliminary," I said. "I want to stress that. We need replication and outside review." "Replicate where?" Patricia asked quietly. "If word gets out and someone publishes first, this isn't just a scoop. This could be exploitation. It could be extinction." That was the line that split the room. Some, myself included, felt the historian's compulsion to place the specimen in the literature. Some, mostly younger and more idealistic, wanted open science, release the sequences, deposit the data, invite peer review. But there was a third, quieter argument forming: protection. If the beings represented a small, fragile population, public exposure could be the death sentence. At 03:12 the next morning, as I was rerunning alignments to be certain, the phone on my bench buzzed. It was Rebecca. She was a medical student back then, years into her rotations, and when my voice came through the speaker, she could hear the exhaustion, even if she didn't see the bench. "Dad," she said, "are you okay?" The voice of your child in the middle of a night like that is a good brace against hysteria. I told her what I could. I did not tell her everything. Parents never do. Even scientists have limits. She said, "Whatever you do, don't be reckless." There wasn't more to it than that, a daughter's moral shorthand, but it was enough to steady me. Later, when I had to justify decisions to others, I would remember the simplicity of that counsel. By the end of the second day, the word Bigfoot had not been spoken in the Institute. We documentarians of the genome felt the gravity without the need for tabloids. We continued the work that morning: deeper sequencing libraries, longer reads, and external controls. I drove a batch of amplified fragments to the university core that had a different platform, partly to rule out instrument bias, partly to create an evidentiary cushion.
There is no romance in the mechanical acts of science at 2 in the morning. It is repetition, pipetting, labeling, checking your peers' math. It took us 48 hours, non-stop, to be confident in the emergent picture. We numbered our slides in a small ledger. We stamped them with confidential. There is a particular hush to a lab making discoveries. It's not silence exactly, but an intimate concentration, like people listening to something fragile. Then came a subtle but unmistakable shift, a call from outside the circles I usually orbit. I got a voice at the end of a secure line that said simply, "Agent Richard Cole, U.S. Fish and Wildlife Service." He didn't introduce himself with ceremony. He had a gravity in his voice that suggested a job where people sometimes lied, and he had to accept the lie anyway. He told me they had seen a report of unusual biological material in my region and requested, politely, that we refrain from releasing any data while they coordinated an assessment. "You're not under subpoena," he said. "Not yet, but there are protocols." "Protocols are for labs," I replied. "We have preliminary data. We need peer review. We need to get this right." "Doctor," he said, with a patience the government affords when it wants cooperation more than argument, "this is not just a lab matter. If there's a population out there, public knowledge puts them at risk, and there are biosecurity concerns on the other side. When a federal voice uses the phrase biosecurity concerns, you are in the middle of an institutional translation. Science's questions have become the state's problems." Agent Cole did not threaten, but he was clear. There would be oversight. There would be a chain of custody. There might be restrictions on publication. He asked that we hold samples under sequestration while they assessed the situation. Holding samples in sequestration is different from sharing them. It is an act that sits halfway between stewardship and surrender. The legal counsel in our meeting, a woman named Sarah Martinez who smelled faintly of citrus and competence, advised caution. "You need documents," she said. "You need controlled transfers. You don't want to hand over material without guarantees." I have a regulator's respect for paperwork. It can be used to protect as well as to coerce. We drafted a receipt. Patricia signed. I personally handed over duplicate aliquots to an agent wearing a service badge and a face like an office clerk. Inside me, something clicked. The Institute's decisions were no longer ours alone. What I did not expect was the leak. Somewhere in those first days, the scent of the specimen found a nose. An obscure online forum posted an unverified note from a source in our state. A local reporter called for comment. Within 48 hours, a rumor had mutated into a headline at a small paper 2 towns over. "Scientists confirm discovery of large primate in Northwest." It read in a tabloid tone that suggested neither respect nor restraint. The article quoted an anonymous source, but named our town implicitly. Our institute's phones lit up like a holiday tree. Patricia fielded the first wave with professional skill. I took the second, a reporter with a voice half cornflower and half concern, asking whether the sequences showed any sign of being human. "I am a scientist," I said. "I cannot comment on speculation. We are analyzing. We ask for time." Time is not always a commodity you are allowed to hold. Time had been dispersed against us by someone who believed that the public had a right to know, or by someone who had a different moral calculus entirely. The leak accelerated everything. It turned internal debate into an external hazard. Agencies began to circle. 2 days after the leak, a compact black car idled outside the Institute. An official from the CDC, a man introduced to me later as Dr. Robert Hriss, came in with epidemiological questions. His presence etched the public health dimension into the problem. If this lineage was susceptible to human pathogens, then an uncontained contact could be a vector for mutual harm. He asked me for contamination controls, for negative controls, for a list of personnel who had access to the specimen. The questions were clinical, but the implication was visceral. They were asking me to list the bodies who might be at risk, even as they implied the specimen could be at risk from us. I remember the meeting in a certain clarity. The fluorescent projector washed the conference room with a blue cast. Dr. Chris had a slow, deliberate manner and a capacity for numbers. Agent Cole was there by his side, practical and unrumpled. Sarah Martinez took notes and asked for legal clarifications. Patricia sat with her hands clasped, and I felt the burden of our lab's name as much as my own. Between the public, the press, the federal agencies, and the simple moral fact of a being that might think and grieve, we were a fulcrum. Officially, Dr. Chris said at one point, we cannot rule out cross-species vulnerability. If the population is small and lacks HLA diversity, exposure to common human respiratory viruses could be catastrophic. He didn't raise his voice. He didn't need to. Words like catastrophic hang in a dry room and settle into conscience. It was right about then that I made a choice I hadn't planned to make. I could have pushed for rapid publication, insisted on immediate peer review and open data. That would have been the scientist's path, bright, public, and accountable. Instead, for reasons that combine the professional and the personal, I began to lean toward protection, controlled data, limited access, and a priority on the living individuals over the paper record.
There is a moral grammar to that decision. To publish is to reveal, and revealing can be an act of violence if power flows to those who would exploit rather than protect. For a moment, I thought of fieldwork I had read about in anthropology texts, documentaries where a pristine culture is made into an exhibit and then collapses under the weight of curiosity. We had a chance to avoid that outcome. The question was whether we had the moral clarity to choose it. Not all of us agreed. At 2 a.m, with another alignment job running, a younger postdoc in my lab said to me bluntly, "You have to think of what science stands for. If you withhold, who are you to decide? Maybe publication might bring the funding needed to protect them." The postdoc's idealism was both admirable and naive. Money and attention do not always arrive as medicine. Sometimes they arrive as guns and trampling boots. That night, as I walked alone on the Institute's back stairs to get a cigarette, an old habit I had never fully shaken, I thought of the moral line between whistleblowing and white lies for preservation. It's an ugly trade to contemplate, and uglier still to make manifest. Before I could settle the issue, something else made the decision for us. On the third night after the specimen's arrival, 2 days after the leak, 3 days after the refrigerated crate, a security guard came to me with a trembling USB and a set of still frames. The security camera outside our cold room had captured a low-resolution sequence at 02-1742. The file was shaky and grainy, shot in monochrome. There was in the frame the shadow of someone or something approaching the delivery bay. We watched the footage in silence. There is an intimacy to viewing a thing on a screen. It compresses distance, but also keeps you a voyeur. At first, the motion could have been a raccoon, a drunkard, a forced-open garage door. It was none of those. The figure stood, bent at the shoulder, then reached toward the crate. For a long frame, decades of biology and a lifetime of skepticism fought in my chest for the proper reading. Then the figure placed what looked like a hand against the metal and lingered, touching it. I remember my mouth going dry. Patricia swore softly. Agent Cole went pale and sat forward. The unknown reader might shrug at the low resolution, at the possibility of misinterpretation. But if you work with sequences for a living, you know that patterns are everything. Patterns tell you what is more likely. The pattern on the screen suggested deliberate, careful contact. Not scavenging, not curiosity in the lighthearted sense, but a human-like attentiveness. We could not comfortably separate lab object from person anymore. We had a body in our custody, and we had evidence that others of the same sort might pay attention to the body's removal. That image, a creature standing in the rain with a hand on a crate, lodged itself in the room like a witness.
There are protocols for this sort of thing. There are forms and chairs and cool-headed men with clipboards. There are also people like Dr. Margaret Chen. I first met Dr. Chen by a late-morning phone call. Her voice was precise and warm at once, like someone who has read too many maps and still loves them. She had spent the previous 27 years listening in a different way, ethnography, not genomes. She told me, in conversational cadence, that she had been working with elders of the Lummi and Coast Salish peoples for decades, and that their oral histories contained steady references, couched in their language, to beings who moved like us and yet unlike us. She named footprints, cairns, and stories of mourning. "We do not tell these things lightly," she said. "There is a stewardship in our telling." There it was, another kind of evidence. Not sequences, not karyotypes, but memory.
And memory, in the hands of people who have been on the shorelines for generations, is a form of archives we are too quick to dismiss. Dr. Chen offered to come up with her notebooks, her cassette recordings, yes, tapes in 1995, and a map full of pins. If what you have is what you suspect, she said, you must speak to us before you decide what to do. We have protocols for these things. The thought of an elder telling me the rightness of restraint humbled me more than any federal agent had. It was not so much that they had authority, it was that they had been stewards longer than my science had been policy. The ethical burden began to tip heavier in a different direction. That evening, the room felt smaller. We had data that suggested a deep relatedness to humans, a chromosome complement that challenged our assumptions, telomere signs of a population bled out by lack of diversity, and security footage that suggested kinship. We also had a leak that would not stop, federal agencies circling, and now an elder's archive pressing for consultation before action. I slept poorly. There is a certain loneliness in carrying a secret this big. It sobers you like bad wine. I kept waking and listening for the humming of the refrigeration units, for life continuing in the world outside the room without us. I tried to imagine the creatures themselves, not as monsters but as beings with histories, perhaps grief, perhaps language of their own. The possibility of personhood made the decision morally significant in a way numbers alone could not. When I returned to the lab the next morning, Agent Cole was waiting. He looked less like an office clerk and more like someone who had sat through many bad meetings. He produced an official-looking envelope with a stamp from a regional office and a line I will not forget. We require custody of all samples classified as non-human hominin until a federal assessment is complete. His wording felt opaque on purpose. It left room for bureaucratic flexibility.
Patricia, who had been roped into dealing with the political side of scientific life for longer than I had, wanted us to hand over the core samples. She feared a break-in, feared vigilantes, feared the press. I wanted permutations, redundancy, peer review. Sarah Martinez insisted on legal language that would protect the Institute's intellectual property and our staff. Dr. Hyris wanted a list of contacts and an epidemiological containment plan. Agent Cole wanted cooperation. I signed off on a controlled transfer of a portion of the sample while retaining others within our sealed cold room. It was a compromise. Compromises are useful, but they are also slippery. On the paper I wrote, partial transfer, duplicates retained by Northwest Institute for Comparative Genomics. Agent Cole read it and nodded, but the look in his eyes told me that paper is only as strong as the signatories who are willing to defend it. As the envelope snapped closed and Agent Cole folded the file into a briefcase, I felt the story tilt towards something irreversible. We had crossed from discovery to custody. The difference is more than administrative. Custody implies responsibility for what happens next. I do not pretend that I foresaw the full sweep of what followed. I only know that the choices we made in those nights, what to share, what to hold, who to consult, set a path that I would wander down for years. For now, the specimen rested in the cold room, and our logs grew into a small library of printouts and anxieties. The nation outside our doors began to murmur. The world, for me at least, got louder and angrier and more complicated. If you have stuck with me this far and want more of the slow, procedural story, how the genetics unfolded, how Dr. Chen's field notebooks added human context, how the federal meetings resolved into legal pressure, and how the security footage later revealed something that would change me forever, then subscribe and stay with the account. There is more to tell, and I will not adorn this with sensationalism. What I will give is the kind of detail I always respected as a scientist: dates, methods, and the honest decisions that followed. What I did not expect was that in the days after Agent Cole left with his sealed samples, the leak would generate something worse than curiosity. It generated a hunger. The next morning, our institute's front door rattled with demands, our inbox filled with FOIA requests, and a television crew staked out the street as if we were a zoo. The attention was not just a nuisance, it was a threat. Whatever moral advantage we might have had in secrecy was evaporating under the harsh light of public appetite. We tried to keep the narrative out of the papers, but stories have their own engines. Someone connected a few dots, and people filled in the rest with imaginations eager for spectacle. Our control over the flow of information was slipping like ice under boot.
That night, the 5th since the specimen's arrival, a senior official from a federal office I will not name for prudence called Patricia and demanded an emergency in-person meeting. They cited national interest and potential public health implications. When you are told something is in the national interest, you know the conversation will move from ethical nuance to legal compulsion. I remember standing in the lab doorway while Patricia and Sarah went into the meeting. I watched the fluorescent reflection on the tile floor and felt, for the first time, an instinctive obligation to the being in our cold room that had nothing to do with career or curiosity. There was a life there, if not a life in the active sense, then a life story inscribed in molecules and behavior, and we were the stewards of its remains. I do not want to claim heroism. I did not plan a campaign of protection. I made small decisions in the dark that in aggregate would define me for the next decade. A retained aliquot here, a cautious email there, a lone call to Dr. Chen to enlist cultural stewardship before public reckoning. When Patricia returned from the meeting, she looked tired in a different way. She closed the conference room door behind her like a person putting a lid on something fragile. "They are not satisfied with partial custody," she said. "They want full access. They are proposing a gag order to prevent any employees from speaking to the press or publishing anything without federal approval. A gag order is a formal method to manage narrative. It is also a form of control that can protect or silence depending on who wields it." I remember the silence that followed Patricia's words like a held breath. For professionals, gag orders are an excruciating compromise between legal safety and intellectual freedom. For the creatures at the center of our debate, it might be protection. I found myself at a moral hinge. The question was stark: publish and risk exploitation, or accept constraints and risk sacrificing the scientific voyage to silence. I thought of the security footage of the hand on the crate, of Dr. Chen's elders who expected discretion, of the telomeres and the HLA profile that read like an obituary in code. And then, as if the room needed one more scoreboard to measure, the fax machine coughed up a page with a photograph attached. It was from a field contact of Dr. Chen's, a black and white printout showing a high, sloping trail in the Cascades, 2 cairns nested like sentries, and at the margin, a blurred silhouette that might have been a person or might have been the play of light on lichen. The photograph's caption read, "We have always known they mourn." There are moments when science and story meet, and neither one is enough on its own. The photograph, the tape, the telomeres, the karyotype, together they formed a collage that insisted on an ethical answer. We could not treat the specimen as mere data.
To do so would be to erase the human pattern of grief that another form of evidence had preserved. Outside, the world kept talking. Inside, we argued, signed papers, and tried to imagine compassion as a policy. The next steps were unclear, but inevitable. Meetings with federal oversight, legal drafts, and the hardest conversations about what to do with a creature whose DNA said cousin and whose telomeres said endangered. I closed my lab notebook that night and wrote, in a cramped hand, a line I would come back to during sleepless stretches for years. If discovery kills them, what is the point of knowledge? The question haunted me as much as any alignment discrepancy. The lab was loud with machines, but the question was a quieter noise. I went home that night and told Rebecca that I was tired. She said nothing more than, "Do what you think is right, Dad." Her trust felt like an overture. When I returned to B Institute the following day, a delivery awaited me, a plain cardboard box with a heavy padlock. Inside, beneath blue-printed wrappers and a note written in a hand that trembled with age, were Dr. Chen's field notebooks, pencil sketches of footprints, cassette labels with dates, and a folded map with pins that marked places the elders had named for generations. The note said simply, "We told you this would not be easy. Thank you for listening." Listening in that moment felt like a moral imperative that outweighed my urge to publish. The data were screaming for amplification. The people with generations of history were whispering restraint. I sat at my bench, the notebooks open, and realized the rest of the week would be a negotiation between institutions, ethics, and the biology itself. There is more to the story: legal maneuvers, meetings that smelled of disinfectant and tension, decisions that sounded small but held consequences larger than our institute budget. There are months in which we cataloged, in which we called elders, in which we sat in conference rooms and argued as if the lives in question depended entirely on the next slide. There are also images that would come later, grainy and moving, that would force my hand in ways I could not have predicted. For now, the specimen lay in our cold room, the labs hummed, the cameras watched. The world outside had an appetite for spectacle. Inside,
a small group of us started to learn restraint. The real shock, I would later come to understand, was not only in the similarity between the genome on my monitor and ours, it was in recognizing how fragile the line is between knowledge and destruction. I tell you this because the choice we faced, to publish and risk exposure, or to protect and risk professional ruin, is not an abstract ethical exercise. It was, and remains, a real dilemma with real living consequences. I made choices that night and in the days that followed. I am still living with them.
When the federal representative called again 2 days later, the conversation would sharpen into a formal demand. When the press outside our doors would escalate from curiosity to pressure tactics, the Institute's leadership would be tested. When an evening security check would produce footage I could not unsee, it would force a decision I had tried to delay. But that is for the next chapter of this account. For now, know what we first found. A body in a refrigerated crate, genome reads that matched ours more than any ordinary primate, a karyotype that suggested a deep but distinct lineage, telomeres that told a story of genetic strain, and a cultural archive that framed the quiet stewardship of people who had been listening longer than modern science had existed. Those findings were the cause of my sorrow and my wonder, and they were the beginning of a moral puzzle from which I would not, in any easy way, untangle myself. When the federal representative called again 2 days after Patricia closed the conference room door, the language sharpened into a directive. National interest, they said. Public health imperative, Dr. Chris said in his even voice. Chain of custody, Agent Cole repeated like a mantra. The cadence of the conversation moved from negotiation to the sort of legal pressure that reads like inevitability. They wanted full access. They wanted control. They wanted the data under federal seal. We pushed back the only way laboratories know how, with documentation. Sarah Martinez drew a line in prose and statute and then built a thin barricade of clauses. She demanded guarantees that any samples transferred would be used only within defined parameters, that personnel would be named and limited, that research would be overseen by a joint panel, including a representative chosen by Dr. Chen and her community. The federal lawyers replied with counterclauses. Somewhere between the two, the conversation became less about science and more about who was allowed to be the steward of a truth that could kill. A gag order arrived as a formal instrument the day after the lawyers began to exchange drafts. It was blunt in its purpose. No employee was to speak to the press, publish, or share raw sequence data without federal approval. The order was framed as a temporary safety measure. That is an elegant way of saying, we will silence you for now, ostensibly for everyone's protection. I signed, because the alternative, refusing at that point, could have meant seizure, threats to staff, or worse. Signing was a surrender and a kind of promise. We would obey the constraint in exchange for some control over how the specimen and its story would be treated. Even signing did not stop the pressure. The military presence increased. Colonel James Patterson, who introduced himself as Logistics and Liaison, arrived wearing the same polite uniform that thinly veiled practical demands. Secure the site, document access, ensure nothing leaves or arrives without clearance. His language was efficient and cold without being hostile. This is not a theatrical decision, he told Patricia during their first face-to-face. If there's an unknown hominin in this region, people will come. They will bring equipment, money, and unpredictable intent. Our job is to reduce the number of variables. When the military speaks of variables, what they mean in practice is perimeter. Perimeter in our town meant more cameras, more badges, and personnel lists printed and checked 3 times a day. For scientists, it meant the rhythm of work changed. We pipetted with eyes at our backs, signed forms before we let a sample out the door, and trained ourselves to describe things in phrases carefully vetted by counsel. Within those constraints, I tried to hold on to the things a scientist should hold: replication, transparentable methods within a closed system, and an insistence on being clear when a claim is an observation and when it is an inference. I continued sequencing when I could, reserving small duplicates, annotating everything in my ledger. The Institute's culture of care helped. We had forensic habits that translated to protection. But even in the sterile routines of lab work, the human elements leaked in. Dr. Lisa Yamamoto, a young anthropologist who had recently become involved through Dr. Chen's network, visited that week. She arrived with a backpack, a recorder, and an urgency in her manner that belonged to a younger generation of scholars who believed that ethnography was rescue as much as study. Her presence was both comforting and unsettling.
She bridged the lived knowledge of elders and the formal apparatus of academia. I grew up listening to these stories, Lisa told me one morning over the hum of a sequencer. They are not mythology the way outsiders imagine. They are living histories, kept in language that resists easy translation. The elders told us of footprints by the salt marsh, of stone piles that are not cairns for hiking, but markers, respect markers. They told us, do not turn grief into spectacle. Her voice carried the weight of someone who had sat on tribal house floors and listened. She said something that landed in me with quiet force. You scientists keep data because it grants you rights in a culture of evidence. Their community keeps memory because it grants them rights in a culture of belonging. Those two rights don't map neatly onto each other. Lisa's intervention reinforced what Dr. Chen had said weeks earlier. Stewardship is not a bureaucratic checkbox. It is a social practice bound by reciprocity. Dr. Chen and the elders wanted to be included in decisions about the remains. They regarded the body not as an object, but as a member of a moral community.
That framing mattered legally and morally. If the people who had tended these places for generations demanded a return, their claim would carry both ethical and political weight. The turning point, what later became the emotional fulcrum of the whole episode, arrived in a file so small and degraded it almost looked like a prank. The security footage had been the first hint that others might notice our custodianship. This later file was different. It was not from our security system. It was from a camera that had been set to watch a narrow clearing in the Cascades, a site pinned in Dr. Chen's map as a place of significance. The tape had been captured by a motion-activated unit that Dr. Chen's field team had installed years before to monitor wildlife movement, not to peer into labs, but to record patterns on a ridge they call it the Hollow. The date stamp on the file read, November 21st, 1995. We watched it in the conference room with only the projector's low light. The footage was grainy, panned across a small stony clearing, and paused on a mound of wet leaf litter. For a long beat, nothing seemed to move. Then, low in the frame, two figures emerged from the tree line, a slow, deliberate approach, bodies moving with a care that suggested ritual more than curiosity. They stopped at the mound. They bent. I will not claim this sequence read to me like a Hollywood scene. The footage was distant and muddled, the kind of image that asks you to assemble the story in your imagination. But there was no mistaking the motion. One of the figures reached and touched the mound. Then the two, carefully, cooperatively, lifted what looked like a bundle and began to move together toward the trees. For close to a full minute they walked in the frame, carrying something between them. They were not carrying a carcass like an object. They carried it like a person, supported and held close to the center of their bodies, as one would carry a child or a close companion in mourning. We all felt the room constrict. Agent Cole swore under his breath. Even the colonel's face softened. Patricia whispered, "They are returning it." The notion lodged like a hard pebble. Whatever had been in our cold room had been, in their language and in their practice, worthy of being taken home. The footage demanded a response that could not be shaped only in policy memos. It demanded a moral reply. We could author such a reply in many ways. We could fight to keep the specimen for study, we could insist on federal custody, or we could, as Dr. Chen proposed, facilitate a return under conditions that minimized human exposure and maximized cultural protocols. Dr. Chen flew up 2 days later. She arrived with elders who preferred not to be named in our logs. They were elderly and deliberate and wore the kind of silence that comes from long experience. In the Institute's conference room, we sat in a circle that included lawyers, a CDC epidemiologist, a military liaison, and a handful of scientists. The elders spoke in short, translated phrases that were clear enough. Our people have been watching. They ask for what is theirs to conduct the care. We do not seek confrontation, one elder said through Lisa, but we will not allow the body to be turned into a spectacle. We will do what our laws require. If you wish to help, be guided by our protocols. The phrase, our protocols, carried weight. They were not protocols of secrecy for secrecy's sake. They were procedures of care. The elders described, through Dr. Chen and Lisa, ways of approaching the body, of not exposing it needlessly to foreign hands, of returning it to a place of meaning, and of marking that place in their customary manner. Sarah Martinez negotiated paperwork that would permit such a return with conditions. Remote monitoring allowed for a specified window. Limited personnel with vetted immunizations and decontamination plans could be present.
All action would be framed as a joint operation with the Institute and federal representatives present in an advisory capacity, and crucially, the elders would lead the handling and decide on final disposition. Those conditions were enough, barely, to satisfy the federal office and to allow a narrow path forward. They still insisted on certain safeguards, decontamination protocols for any human participants, a list of permitted observers, and a requirement that any biological samples taken during the operation be catalogued and placed under federal custody. The elders agreed to those terms with a simple clarity. They would accept scientific caution, but they would determine how the being was treated. The return was scheduled for November 24, a day cloaked in cold and rain. The Institute provided logistics, a low-profile transport that did not advertise itself, personnel cleared by the CDC, and a quiet plan executed before dawn. Dr. Chen's community prepared its rights. We agreed that scientists would not photograph the event in any public-facing way. Our presence would be practical and advisory, not performative. I remember the drive to the clearing like a string of small, resonant images, the van's heater coughing to life, the road shining black with rain, the sound of the tires and wet leaves, and the thought that I was participating in what felt like a homecoming. The clearing itself, when we arrived, was ringed by stone piles and small, simple markers the elders called witness stones. The sky was low. Dr. Chen moved with an economy of motion that matched the elders' dignity. The federal officials were present but placed at the periphery, their badges tucked beneath raincoats. The body, no longer on a sterile aluminum gurney, but wrapped in the elders' cloths, was carried by a small team led by 2 elders who moved with a purpose that was at once ritual and practical.
They placed the bundle at the center of the clearing and then performed gestures that have a long history among their people. An offering of water, a small song, the cadence of which I couldn't follow, and a soft, sustained watching. The atmosphere was the opposite of spectacle. It was close and contained, as if grief itself had boundaries. We set up remote cameras at a respectful distance, as agreed. Their lenses were angled not for voyeurism, but for evidence, for a record that would show how the body was received. We left a distance that allowed the elders privacy, and yet gave enough information for scientists and the federal people who insisted on oversight to know what happened. The cameras were motion-activated and recorded at wide angles. Their footage would later provide the crucial confirmation that shifted the moral axis of the episode. The elders worked in a manner that suggested they had already rehearsed this in their prayers. They moved as if returning someone to a home that had been waiting. When their hands touched the cloth that wrapped the body, there was an audible hush among the assembled scientists and officials. The elders did not ask for permission to grieve. They claimed it as a right. They spoke aloud in their tongue, and then, for our benefit, whispered a short translated line through Dr. Chen. "We take her back." The pronoun, deliberate, humanizing, settled in me with a force that made the rational language of karyotypes and telomeres seem abstract and secondary. We withdrew to the perimeter and watched the remote monitors discreetly. The footage, later reviewed in more durable form in our lab, was grainy, wide, and from a distance of 30 to 40 meters. What it showed was not a parade, but a simple series of movements. 2 figures moving as a pair, a pause, an orientation of faces to the mound where the body had been placed, and then a moment that I will not forget. One of the figures reached out and laid a hand, not a quick touch, but a long, considered placing of palm, against the wrapped bundle. The motion was the same care we use when closing a child's hand around a toy. The companion then gathered, and together they lifted the bundle and began to walk away down the path into the forest. The scene contradicted any hypothesis that these beings were spectacles or monsters. They were acting in a way that signaled relationship, mourning, and responsibility. To anyone with the capacity for empathy, the footage was evidence not of a sensational other, but of kinship and practice. It was behavioral confirmation aligning with what Dr. Chen's elders had described in words and photographs. They mourn, they carry, they return. That the footage existed and that we were witnessing it in the months after the specimen had arrived changed everything for me personally. The question transformed from an abstract taxonomical puzzle into a mortal ledger with human faces.
I found myself trembling in a chair in a way that no lab error had ever provoked. I do not believe I wept openly, not there. We were all professional actors in a scene that required stoicism. But afterwards, alone in my office, I allowed myself a quiet collapse against the chair, and the tears came for reasons that had nothing to do with career and everything to do with human empathy. The federal offices, faced with the footage, adjusted their stance. Public health concerns remained salient. Dr. Chris reminded everyone of the HLA findings, the telomere data, and the susceptibility that population genetics had suggested. But the presence of witnesses and the cultural claim complicated any move toward open exposure. Who would we expose? The beings who had come to retrieve their kin, or the elders who had asked for discretion? Public justification for unilateral seizure weakened when confronted with the elders' claims and the optics of oppression. The resolution that emerged was messy and negotiated. In legal terms, it was a modified custody arrangement. The Institute retained sealed copies of certain data to be archived in a secure facility accessible only to a panel we helped assemble, including 2 representatives chosen by Dr. Chen's community, 2 federal scientists, and 2 Institute scientists. Any plan to publish identifiable information would require unanimous approval from that panel, an arrangement that protected the beings from hasty exposure while preserving the possibility of controlled ethical scholarship in the future. In return, the elders agreed to certain steps that would protect public health. They permitted a small, vetted team to remain for a limited observation period. They accepted decontamination protocols for scientists who might enter the area, and they allowed the remote cameras to run for an agreed window. There was no contract worthier than the elders' word, but paper was necessary for the federal systems that needed records. The modified gag order went into effect as part of the settlement. It was no applause for secrecy. It was a legal instrument that bought time. For the Institute and for me, it also bought something else, the preservation of life. I signed the order with reluctance and conviction in equal measure. I agreed to hold back certain data from publication to prevent a stampede of people poisoning the habitat either by curiosity or by some misguided philanthropy. The days that followed were an odd mix of relief and continued tension. We secured the archived data. Agent Cole and Colonel Patterson ensured the perimeter had sufficient coverage without turning the site into a military outpost. Dr. Hriss issued guidelines supposing worst-case scenarios. Dr. Chen organized a slow, patient monitoring regime. My nights were still short, but no longer the ones of frantic analysis. They were the nights of a different kind of vigilance, where the aim was preservation rather than discovery.
I continued to record everything in my ledger because the scientist in me could not help itself. The telomere curves, the karyotype photographs, the HLA heterozygosity figures, each entry was annotated with the phrase, "retained copy," archived under panel custody in my cramped handwriting. These records became my private religion, careful, precise, and ultimately a confession. Years later, in interviews that could not be conducted publicly at the time, some asked why we had not published in Nature, Science, or other mainstream journals. My answer was always the same and simple. Publication without protection was a moral irresponsibility. We had, in our hands, the power both to illuminate a deep biological truth and to destroy what light we sought to reveal. The choice to delay was ugly from an academic perspective. It was raw, antithetical to the impulse toward credit and precedence, but it was right. What followed the immediate crisis was a long, low-motion project of stealth protection. The elders and Dr. Chen's network implemented soft conservation measures that were obfuscated publicly as habitat restoration projects, fisheries enhancement programs, or cultural preservation grants. Federal funds were quietly redirected into land purchase and corridor creation around the places the elders had identified. The initiatives were not billed as Protect Bigfoots. They were fenced under the bureaucratic IDs of wetland preservation and tribal cultural sites. The rhetoric was conventional, but the outcomes were protective. My role evolved into that of an uneasy guardian. I was still a scientist, running assays, annotating sequences in my locked notebooks, but I was also a participant in a social compact that stretched beyond credentialed expertise. I signed documents. I advised on decontamination. I advised on who could be invited into the fold and who could not. I even began to think about the long-term preservation of knowledge in a way that did not put living beings at risk. Part of that strategy was cunning in a modest sense, to encode the science into forms that would not, if leaked, serve as a map to a physical location or a hunting ground. I wrote a book. I say wrote loosely. What I authored was a hybrid, a speculative narrative that folded technical detail into a fictional frame. It was crafted to communicate the ethical stakes, to preserve the sense of wonder and danger, and to convey the science in a way that could be scrutinized intellectually without revealing specific coordinates, samples, or exact sequences that could be misused. I did not call the book Proof. I called it The Hidden Cousin, a title that captured the awkward intimacy of the relationship without furnishing a GPS. The publisher insisted on a certain marketing arc. I insisted on a clause that barred release of any raw sequences or identifying data. The manuscript was published as a novel with scientific rigor layered beneath the fiction.
It was, in effect, a cipher. Anyone reading carefully could find the ethical argument, the reasoning, and the conceptual frameworks. They could not, from the text, reconstruct a chain of custody that would lead to fieldwork that might harm living beings. The book was controversial in academic circles. Some colleagues accused me of cowardice for not publishing the raw data. Others thanked me quietly in letters for preventing a calamity they had not known how to name. It was imperfect, and I own that discomfort. But it was a way to keep the conversation in the public sphere without operating as a target. The project of guarded stewardship had costs beyond the professional. It demanded secrecy. It demanded complicity. It demanded I accept that some truths would be held close and not waved like flags. It also demanded reparative work. I attended forums with tribal elders, learned aspects of stewardship practice I had never imagined, and slowly, haltingly began to understand how to translate my scientific impulse into a framework of moral preservation. Years folded on this axis. The remote cameras ran intermittently for months. They captured what the elders had said they would: movement patterns, seasonal migration points, evidence of tool use in the form of broken branches arranged in the way the elders described and not as natural decay. One clip showed, in late spring, a small structure of bundled bark arranged in a way that looked deliberate. Another recorded a group rhythm, people passing a task from one to another in a coordinated, social action. These were not proofs in the mathematical sense, but they were corroborative behaviors that aligned with cultural stewardship. Over time, the federal urgency dulled into bureaucratic steady state. The panel's oversight rotated personnel, funding lines shifted, the perimeter returned to its normal legal geometry. The project that had once drawn a swarming press corps became a small ledger entry in several offices. For me personally, the moment of greatest clarity came not from a graph or an alignment, but from a conversation with Rebecca. It was late spring 1998, and Rebecca had a little boy then, Marcus, who liked to look at microscopes even though he was too young to hold a pipette steady. He would press his nose against the stage and call tiny things friends. On a quiet afternoon, she visited the institute with Marcus perched on her hip. He toddled curious and stopped at a sealed box I kept in my filing cabinet. It had my notes inside, printouts, hardened aliquots in legal storage, and the tiniest of everything that could be used to reconstruct a sequence if someone had both access and intent. He pointed at the box and said, in the blunt language of children, "Grandpa, what's that?" For a moment, the scientist's response, classified, sensitive, rose in me. Then Rebecca looked at me and said only, "He's yours as much as he is mine. When your time comes, what will you leave him?"
Her question was not rhetorical. She wanted to know what legacy I would hand Marcus: a ledger of secrecy, a moral lesson, or something else. I told her the truth, that I could not allow everything to be public, because publication might be a sentence for the living. I said that I needed time, and that time was not something I could manufacture forever. She listened, and then, with the practical wisdom that belongs to parents who know the balance between protection and truth, she said, "Then make it survivable. Teach him enough that someday he can understand why you did this, and trust him with the rest." That conversation prepared me for a decision I had been resisting, to set aside a trove of material in a place with precise instructions for future custodianship. I drew up a will-like document that was legal in structure and moral in intent. It named a sequence of trustees: Rebecca, a trusted elder recommended by Dr. Chen, a scientist unaffiliated with the original institute, and a legal guardian with a reputation for protecting culturally sensitive material. The instructions were clear. The archives would remain sealed for a period I could not fully vouch for, but suggested was measured in decades, after which an appointed panel would reassess. The goal was to pass responsibility forward, not to absolve it in my death. I called Marcus my moral hinge, because the concept seemed to fit. He was the next generation, a person who would inherit the consequences of our choices. The archives were not simply about DNA. They were about a story made of science, humility, and restraint. They were an appeal to future judgment. Time moved. The remote monitoring continued in fits and muted bursts. There were summer seasons where the cameras showed little, and others where the footage gave us enough to understand the rhythms of a population small and fragile, as our models had predicted. The HLA patterns we had recorded suggested continuing low heterozygosity. There were no miraculous recoveries. The models of decline, which I had once run on a coffee-stained printout, remained grim. But the actions taken, protective land purchases, reduced human incursion, and cultural stewardship, bought time that models had not accounted for when run without human intervention. Every so often a rumor leaked into the public sphere, usually thin and quickly quashed. A weekend hiker's alleged sighting would ripple through local forums. A camera trap from a neighboring conservation project captured an anomaly and was declared inconclusive.
Each rumor was an oscillation of risk. Each required renewed caution. We learned to treat curiosity itself as a vector. A decade after the refrigerated crate arrived, I found myself sitting in a quiet room in 2005 to sign off on the final legal language of the archive's placement. I was older. The edges of the years had left me with a stooped patience. My hair had more silver, my hands a faint trimmer.
Rebecca had 3-tone gray streaks of experience. Marcus, now 9, listened to my voice with a rapt attention I found both ungainly and humbling. We gathered in a small notary's office. Dr. Chen was present in spirit through a letter she left with the archive, instructions, a blessing, and an admonition to keep stewardship at the center of any future decision. Sarah Martinez, who had become a friend over the years and a fierce guardian of legal nuance, signed in the margins like a soldier at attention. Agent Cole had retired from his federal post, but he wrote a brief note that would later be sealed with the archives, a pragmatic recollection that our actions had likely averted several hazardous confrontations. I deposited in the archive not only the printouts and the copy of the telomere assays, but also a small notebook of reflections, more confession than method, a personal narrative I hoped would explain motive more than technique. The panel's terms were spelled out in legal language intended to survive shifting administrations and institutional memory. The goal was to make stewardship a structural rule rather than a series of ad hoc decisions made under pressure. In 2005, I took Marcus into the lab for a brief visit. It was an arranged moment, low risk, and supervised by people the elders trusted. He peered at a microscope, mispronounced karyotype, and told me with the absolute certainty of a child that he wanted to be a protector of secrets. I laughed, partly out of relief and partly because the phrase seemed appropriate. "Protector of secrets," I said, and handed him a sealed envelope that contained a photograph he was too young to appreciate. It was of the clearing with the stone piles and a faint outline in the distance, the distant protective image that had haunted me for a decade. I told him the photograph was a story he might be ready for when he was older. He tucked the envelope in his pocket as if it were a treasure map. The final years of my involvement were quieter than the opening months had been. I published the novel as a moral gesture. I gave lectures on ethics and conservation framed around theoretical dilemmas rather than any identifying facts. I maintained contact with Dr. Chen and with Rebecca. I lived with the knowledge that I had chosen to shield living beings from a public that tends to be greedy for revelations and clumsy in restraint. The decision had cost me professionally in some circles. It had gained me peace in others. There is a story I sometimes tell, partly as a way of testing my own conscience. It is about the day the security guard brought the shaky file that showed a hand on a crate. I tell it not for sensational effect, but because, for me, that image was a turning point that reframed the ethical problem.
If a creature could stand in the rain and touch a crate with a hand that cupped metal like it was touching something dear, then the device of objectivity that some scientists cling to is insufficient. We are biological beings capable of empathy. The law recognizes personhood in many contexts. Should moral imagination be slower to follow? There are critics who will always claim that we hid data, that science was betrayed by secrecy. They frame the problem as a simple trade-off, knowledge versus ideology. To them, I say this: science exists within society. When the act of publishing itself can increase the risk of extinction for an intelligent lineage, the ethics of restraint become part of scientific duty, not an opposition to it. Our obligation was not merely to know, it was to preserve the possibility of people, whomever they might be, to continue living their lives without being preyed upon by curiosity. I do not offer a clean moral calculus. There are regrets. There are nights I lie awake and imagine alternate choices that might have produced different outcomes, some better, some worse. But I can say with conviction that the choice we made to prioritize living beings over a paper did save possibilities. It preserved habitats that otherwise would have been open to tourism and exploitation. It respected cultural protocols that would stand time in ways policy memos do not. And there is another element that keeps this story from dissolving into simple nostalgia: the obligation to record. My ledgers in the archive are incomplete by any rigorous scientific standard. They were never intended to be a last word. They are a stewardship, a bridge between the needs of science and the claims of moral guardianship. I left instructions for future reevaluation. I embedded within the archive a path that could be followed only with permission and a moral consensus. That is not perfect, but it is honest. When people ask me whether what we found proves the existence of Bigfoot in the sense of tabloid lore, my answer is that labels matter less than behaviors in history. In my analysis, clearly marked as such in the archives,
The genome showed a close affinity to humans at roughly 98.7% similarity by the metrics we used then. The karyotype was 48 chromosomes. Divergence time, modeled conservatively, placed the last common ancestor at about 1.2 million years ago. The telomere GINA and HLA profiles suggested a small, effective population with low immune diversity. Those are the technical facts as I recorded them, always framed as model-based estimates and never as universal pronouncements. The rest, the mourning, the carrying away, the cultural stewardship, are corroborations of personhood and behavior, not declarations of rights. In quiet moments, I sometimes think about the line between knowing and preserving. There are people whose moral compass points towards disclosure at all costs.
There are others whose compass points toward guardianship, even at the cost of reputation. I inhabits both worlds, and learned the hard way how little pleasure either offers in the short term. There is a long twilight where regret and relief coexist. The final image I will share from this sealed decade is simple and human. It is a photograph, taken with consent by one of the elders, and only for the community's archive. Not of a body or a specimen, but of a small stone pile beside the clearing. In front of the pile is a child's sandal, weathered but placed with care. A smudge of lichen looks like a thumbprint. To me, the picture reads like the ending of a letter someone wrote long ago. We were here. We remember. We will keep watch. In the end, the shock that the headline promised was not the revelation of an alien monster beneath our pines. It was the recognition that the beings we found were close enough to be kin in DNA and in behavior, and fragile enough to be erased by exposure. The true shock was ethical, how knowledge can hurt when wielded without conscience. My life since that November has been an attempt to live out that recognition responsibly. I signed papers, arranged archives, negotiated with agencies, and labored to protect fragile lives, because someone had to balance the ledger. Rebecca watched me choose restraint, and taught me, by example and patience, how to live with the cost. Marcus will inherit a story and some documents, and, I hope, a moral instruction about the duty that comes with knowledge. I have tried to tell you this without sensationalism. I have tried to be precise where precision is due, and careful where conjecture would be cruel. This is my account, bounded by dates and by the evidence I recorded. It is not a final scientific verdict for the world. It is a first-person story with the weight of a laboratory ledger and the human choices that followed a number on a screen, 98.7%. If you have found this narrative troubling, that is a sign the trouble was real. If you are angered by secrecy, I ask you to imagine the alternative. If you are tempted to demand all documents now, consider the lives they could expose. There are no easy answers. One last note. The archive I arranged will not be opened lightly. It will require consensus that I can scarcely predict from this vantage. In the meantime, there are people, elders, scientists, and a next generation like Marcus, who are willing to hold the responsibility and to watch. That willingness, more than any paper, was the gift of those years. If this account raised questions about science, ethics, or conservation that you'd like to explore further, please like and share. We have a playlist of deep dive episodes on science mysteries and ethical dilemmas that follow this same thread, questions about what we should do when knowing is a danger rather than a benefit. If nothing else, remember this. Sometimes the highest duty of knowledge is to hold its tongue.